Hello everyone, great to see you. Great to see that some people in the chat. Sorry, Lewis, that you are in classes and not able to do it later. Katrina, you should probably focus on the work that you're meant to be doing. But great to see everyone, and thanks for joining. It's just something that I thought would be really helpful for, if I was at school and in lockdown and not be able to go to normal school and see my teachers, then I thought this would be something that I'd find helpful to have. Something like an online lesson, something a bit extra outside of school that might help out. So that's the aim of this, and we'll try and do it as regularly as possible, hopefully every week, and we'll generally work through the UK GCSE syllabus. But it's all stuff. The amazing thing about biology is it's universal to all cultures and all languages and all specifications for exams. So great that we can talk about cells, and actually they're the same cells that make up me as the same cells that make up you. And okay, there are different cells in plants, but they're the same the world over. So that's something that I love about biology, so that's why I thought it would be a useful thing to do. And let me just show you what our plan is for today. So we're looking at cells, we'll look at the structure of cells, we'll talk about how they are specialised and look at some specific specialised cells. We'll talk about cell differentiation and briefly about how cells end up from being very boring, normal cells that can become anything into very specialised cells that have a really specific job. And we'll end up with a bit of microscopy as well, just because how do we see cells? That's helpful to know as well. Hi Daniel, thank you for watching, and I'm glad you got to university. And hey, a little bit helps, doesn't it? This is some new technology. I'm using Streamlabs and then writing and stuff, so bear with me if it doesn't quite work, but we'll see how it goes. To start off with, we're just going to look at an animal cell. So really basic and something that you've probably seen before. They have not really got a fixed shape. If we look at a plant cell next to it, then a plant cell does have a fixed shape, and they are much more rigid and often kind of more square. You know what, I'm going to end up drawing it underneath because then at least we can all see it. But let's stick with the animal cell for the time being. So an animal cell, and the first thing we tend to draw is the cell membrane, which is around the outside, and then in the inside of the cell membrane there is the cytoplasm. And the cytoplasm is this jelly liquid. It's actually got different structures inside it as well, and that's going to be where all the reactions take place inside the cell. So everything happens within the cytoplasm, and that's bound by the cell membrane. And the cell membrane itself, that's more of the security guard. If you think of the border guard, that is the thing that's going to control what comes in and control what comes out. We've drawn it really simply with just a line, but in fact there are lots of different proteins that work as channels. Some proteins that even have little motors on that be able to push things through, some that open at certain voltages, some that open only when other molecules are bound to them, and they open to let different molecules in. So they're really complex. It's a really intricate system. It's a mosaic of lots of different things, and so it's a fantastic thing. But we've just represented it with a line, which seems a bit petty, but I'm sure the cell membrane is not offended. The next thing that is often drawn, there we go, is a nucleus. So a nucleus in the middle of the cell, and that is something that's going to have all the DNA, all the genetic information that you will need inside a cell. That DNA is the same in every single cell in your body, and in a plant, it would be every single cell in that plant would have the same DNA, and that what makes us unique. Peter asks, is it live? Is it live? Of course it's live. I am live, and I'm drawing it live as well. Don't doubt me, Peter. Now, other things that are going on inside a cell, because there's lots of interesting stuff. A cell needs to get some energy, and so inside here there are some little mitochondria. And mitochondria, you might have heard of them, an internet meme as the powerhouse of the cell, but actually we can describe it a bit better than that. And rather than saying the powerhouse of the cell, we can say it's the site of respiration. Let me just spell mitochondria properly first. So when we do respiration, what it is just turning oxygen and glucose into carbon dioxide and energy. And so that is what happens in these mitochondria. If we zoomed into one, then it's this kind of oval-shaped structure. It's actually got all these layers inside as well. So it's got a really high surface area, and there's all sorts of complicated things that happen to make respiration happen. And so it's an amazing thing that's inside a cell that means a cell can do it by itself. And actually, it was thought that at some stage the mitochondria was another cell that ended up working with animal cells in general in order to make them work properly. And so the mitochondria might have once been a different type of cell that was inside a human cell and worked with it. And now it's ended up being very, very important, not just in animal cells, but in every cell really, and needs to get energy. Now, another thing that we might draw inside an animal cell is some not mitochondria, we've done there, some ribosomes, okay? And so ribosomes, they're tiny, they're just a couple of proteins fitted together, and they help put together RNA and turn it into proteins. And so they're very tiny, so we just have to do little dots for them. But we can just draw them in there. Ribosomes, fantastic. And that is the basic structures of a cell, of an animal cell. And it looks quite simple, but actually when all these things are working together, then it is quite complicated, and there's plenty going on inside there that is going to make it work together. Now, if we drew a plant cell, we've already said that it's going to be slightly squarer. I tend to say it's a rounded rectangle for a plant cell. And you might have thought that the thing that we've drawn around the outside is the cell membrane, but in this case it's not. The thing on the most external of a plant cell is actually a cell wall. And we tend to draw it as a double layer just to show that it's a little bit thicker, and this is what's giving structure, meaning that the cell will not move. It also means it won't burst if it fills with water. So this is the cell wall. Now, if we look inside the cell wall, then we do still have a cell membrane, because a plant cell still needs to be able to control what comes in and what goes out. And so that is on the inner layer of the plant cell. And the cell wall here is worth noting that it's made of a chemical called cellulose. Now, cellulose is actually just a long chain of lots of sugars, so it's a bit like starch in that case, but in this case the sugars, the glucose molecules, are slightly arranged differently, and so they end up being this really strong structure. So we've got a strong structure for our outer cell wall in a plant cell. Now, it's important that we remember that it's cellulose there, because actually in a different cell that we'll come on to, the bacteria cell, we've got a cell wall, but it's made out of something different. But we do have some familiar structures inside a plant cell. We have our old friend, the nucleus. We also have mitochondria, and we have things like ribosomes. You might be thinking, hang on, they've left a bit of space in the middle. That's because there's often something inside a plant cell called a vacuole, or a permanent vacuole. So this, let me squeeze it around the banner. This vacuole is a storage of sugars and water, often called cell sap, inside the plant cell. And if you ever chopped down a tree or seen the stickiness inside of a leaf, or even inside a fruit, it's that same sort of sugar and water that is inside a plant cell. What you're doing is bursting the cell open, and the sugars and water from the vacuole are coming out. And so that is really important for the plant. It also helps them increase their storage of water. So you think actually plants rely on water to give them structure. They don't have bones, but if you fill up the cells full of water, then they're full. They would be bursting, apart from the fact they have a cell wall to be able to hang on to the cell to keep it in shape. And because it's got that structure, it then means that it won't burst and it can hold on to the water. Great, now there's another thing that is in a plant cell, but not in an animal cell. And I'm just going to draw these slightly differently by shading them in and maybe a bit more circular. Now these are chloroplasts. Now a chloroplast is another organelle, something inside a cell, but this one can do photosynthesis. And so the mitochondria we talked about, they are a little thing that does a big reaction, a big important reaction. This is another little thing that does another big important reaction. Photosynthesis is something that turns carbon dioxide and glucose into water and oxygen. I feel like I've got that the wrong way around. I have. So it turns carbon dioxide and this time water into oxygen and glucose. I'm going to write that down so I don't forget it. So photosynthesis is, in this case, carbon dioxide, CO2, plus some water, man, I'm all over the place, goes to glucose, which is C6H12O6, and also some oxygen, which is O2. It's good to get these things right. Now, we want to make sure this equation is balanced as well. Let me just scooch it down so you can see it. And so if we've got 6 carbons on this side, we need 6 carbons on this side, so we can put a big 6 there. Now, if we count up, we've got 12 hydrogens there, and we've got an H2 there. We can't separate those hydrogens, and so we need to put a 6 there, so we get 6 times 2, that's 12. And then on this side, hang on, now on this side we've got 6 times 2, that's 12 oxygens, and another 6 there. So in total we've got 18 oxygens on this side. We need to have 18 on this side. We've got 6 and we've got another 2 there. If we put a 6 there, then we've got our 12 plus another 6. We've got 18 on both sides. So this is our perfectly balanced equation for photosynthesis. If you're stuck, it's always worth trying to write something down so then you know what's going on. That is our plant cell. It's a little bit more complicated than an animal cell in the sense that it has photosynthesis capabilities. It's got a vacuole inside. It's got a cell wall made of cellulose. But other than that, it's just got the basic structure of an animal cell with a few added on. If we go on to something completely different, like a bacteria cell, then actually it has some of the same structures, but in quite a lot of them are different, and it doesn't have some structures that an animal cell and a plant cell both have. So we tend to draw these again as kind of like a rounded rectangle. I tend to draw them on the side a little bit because then they're going to move in one direction or the other, whereas plant cells don't move as much. And they have got a cell wall again, but as I mentioned earlier, the cell wall inside around a plant cell is made of cellulose. This cell wall is made of a chemical called chitin. CHITIN. And so it's just a different chemical that they use to build a cell wall. It's actually something that we use with antibiotics. We target bacteria cell walls because this chemical chitin doesn't really turn up naturally in a human body, but it does turn up naturally in bacteria. And so we can use an antibiotic that will target a chemical that we're not going to be using. It doesn't matter if we block this chemical or block the production of this chemical. It's not going to damage our bodies, but it will damage the bacteria and hopefully kill them. So that's why we often will target something like chitin, or even just the molecules that make the cell wall out of chitin in order to use an antibiotic. Now inside, the next often thing we draw is a cell membrane. So again, it's there, just on the inside of the cell wall. And we might often then think about a nucleus. However, this time there's no nucleus. That is the big difference between a bacteria cell and a plant cell. But what they do have instead is this kind of loop of DNA. And it's just a little bundle. There's nothing wrong with them having that instead. It often means that their DNA is a little bit shorter, but it does still do the job and all sorts of things. I've seen a few people asking about the structure of the brain. I'm happy to do a little bit about the brain. I did do a short video on one about no blood in the brain, so do go check that one out. I'm not going to touch on it today though. We might talk about nerve cells, and you've got lots of nerve cells in the brain. But yes, hang on in here. If you subscribe, and then you'll see all my videos, then I'm sure you'll be educated on the brain in no time. But back to our bacterial cell. So we've got our loop of DNA here. And The thing about this is it also has other bits of DNA in as well. So it has little circles of additional DNA that are separate from the first bit. So there's a big loop of DNA for all of it, and then the little loops of DNA are called plasmids. Now, someone's just asked if we're doing specialized cells such as neurons. We will. It's in the plan. If I show you the plan again, I showed you at the start. We're on structure at the moment. We'll talk about specialized cells next, and I'll make sure to include a nerve cell in there just for you. So these plasmids, these are really interesting. So it's a circle of DNA that contains information for just a couple of genes. And so normally you think, well, where do my genes come from? Where did you buy genes from? No, different genes. Where do you get your genetic information from? You get it from your parents. So it gets passed down from your parents in a sperm cell, an egg cell, it fuses, and then you've got those genes and they're yours for the rest of your life. Bacteria cells do that as well, and they split and they pass on their DNA to some their offspring, but they have another form of passing on their DNA, which is that they can actually pass it between different living bacteria. So it's a bit like me going up to someone and saying, hey, I've got brown hair, do you want brown hair? Let me give you the gene for brown hair. And somehow extracting this gene from one of my cells and inserting it into one of their cells. And then they have brown hair. And so the bacteria can work like that, and it's a bit weird if you imagine it like that, because we're so used to just genetics passing down from family to family, from one generation to the next. You can't pass your DNA to a brother or a sister or a friend or anything like that. But these DNA inside a bacteria cell, they can unravel this little plasmid, poke it out into the next door cell, and then they can make a copy of it and have it for themselves. And it's a really important thing for bacteria to be able to have characteristics to protect themselves. So for example, bacteria that have antibiotic resistance might just have a little plasmid of antibiotic resistance, and they can pass that on to their neighbouring bacteria cell, and suddenly all the bacteria have antibiotic resistance. So great for bacteria, less so good for us and for doctors trying to deal with bacterial infection and trying to treat them with antibiotics, but it is a pretty cool way of passing on your DNA. Now, back to this cell, and there are a few other features as well. Obviously, it's got cytoplasm inside there, and it will most likely have ribosomes as well, although they're slightly different in a bacteria cell. I think they're slightly smaller. And they can have photosynthesis capabilities as well with chloroplasts. They can have mitochondria. The last thing we'll add, though, is a flagellum at the end here. This is how they propel themselves forward. Oh, you can't see that, sorry. And so the flagellum are these fixed structures, a bit like you'd see on the back of a sperm cell. Often we think of them as kind of wiggling a little bit, but instead they're a fixed structure that then rotates, and that propels the cell forward. And so the bacteria cell will probably have some of those to be able to push it along. And another special gross bacteria cell feature is that it might be encased in a kind of capsule or have another layer, which is like a slime layer. And so that just means that these bacteria cells that normally are cells by themselves, they can group together, they can stick together, and so they can end up forming a big blob of bacteria cells, which means it's easier for them to pass on plasmids to each other, and it's also easier for them to fight off something like a white blood cell that's coming in trying to destroy them all, because they can then work together and release some toxins and cause some damage. So they've got a few different features that help them do that. Great. So I would say that is the basics of cell structure. So they're kind of key features that turn up in every single cell. So we can tick that one off, and I think it's worth moving on to specialized cells. If you do have any questions, keep putting them in the comments, that's fine. I'm happy to answer them as I go, rather than taking questions at the end or something like that. So that's absolutely fine. So great. The next thing we're going to talk about is specialized cells. So we're going to look at some specific cells, and the first one I thought we could look at is a red blood cell. Red blood cell, I thought I'd change colour to red because it's important to have some variety and things like that. So we draw a red blood cell by, it's a circular cell again, but often if we just look at the outside of it, it's this circle which has a little divot in it, So you might think of a donut, like a ring donut. It's got a hole in the middle. Red blood cells like a donut, except instead of the hole in the middle, it's just got another bit of cell membrane. And so if we looked at that, if you think of that donut like that and you turn its side on, then in a red blood cell it would look like this. And it would have a kind of divot in the middle. And so this is a concave surface. And a concave surface just means it's one that's curving in on itself. If it's convex and it's curving out on itself, a bit more like a sphere. And because it's got two of those, we'd say this structure is biconcave. And then because it's this circular shape as well, this kind of donut shape, we say it's a biconcave disc. And so this red blood cell has a biconcave disc shape, and the reason for that is that then it can have a large surface area. And you might be thinking, well, why do we need a large surface area? We've got to think back to what is the job of a red blood cell. Red blood cell's purpose in life is to be able to carry oxygen around the body. And so if it's got a large surface area, then it can more easily pick up the oxygen in the lungs, and it can more easily deposit oxygen where it needs to. So all of its different adaptations are all to do with that job. And so large surface area, so it can pick up loads, and so it can dump loads. And the next thing that it has, or rather doesn't have, is it has no nucleus. And so the fact that it has no nucleus means that it's got lots of room still for oxygen. But instead of having lots of room for oxygen, it actually has lots of room for the protein that carries oxygen. So from what I mean is lots of room for the protein that carries oxygen, and you might know the name of that, that's called hemoglobin. And so hemoglobin is this protein that has 4 parts of it that can carry 4 oxygen molecules. It's got hemo because that's to do with the iron. So when someone's anemic and they might look pale, that's because they don't have enough iron in their blood. And so iron is in the red blood cells, and that makes your skin look red, and or your blood look red even, and things like that. We've got a couple of questions, so let me just pause briefly. Can I explain the new COVID vaccine in terms of the mRNA technology behind it? Yes, absolutely I can. I might just finish this cell first and then we'll get on to that. Is that only in the middle where you cut it in half, or does it look like that on the side no matter what? So it's a bit like, let me just explain again because I think that might help. If I chop this cell here down that side, then it will look like this. If I chopped it, so you imagine a donut, obviously if I chopped a donut like down there, it would look like this again. And so, yeah, imagine my donut's like that, I'm chopping it down like that in order to end up with this shape here. Now there are just lots of arrows, and it probably doesn't make much sense. But oh well. The third thing that I want to tell you about red blood cell, and then I'll briefly divert about the COVID vaccine. We'll just turn the pen off. So it's biconcave disc, we say it's no nucleus, and then we can also say that it's got this molecule, hemoglobin, that binds to oxygen. And it's actually a really, really clever molecule. So it binds to oxygen really easily in the lungs where there's lots of oxygen, but when it goes to somewhere that really needs the oxygen, so then if it goes to something like a leg, that's when you're going for a run and the leg's using lots of oxygen to make lots of energy, then it suddenly doesn't like hanging on to oxygen very much and just gets rid of it. And so that kind of combination of being able to really grab onto it really easily when it needs to grab onto it easily, and really let go of it easily when it needs to let go of it easily, is a really fantastic property about it. So it's a good one. Let's just pause. Someone's asked if this lesson will be on the channel for reference. Yes, don't worry, it will be. I thought also I'll put these notes on as a PDF so you can see them and you can have them, because they're helpful, and then you can look back at them and try and decipher my writing without me explaining them. Let's just go to an aside in a blue pen over here and talk about mRNA and the vaccine. So And the way that COVID works is that it gets into your body. It's a virus, so it enters a cell. It can't reproduce by itself, but it enters a cell, and it does that by having little markers on it that make your body think that the cell is part of the same cell, so it goes in quite easily, and then it starts churning out more. The cell basically turns into a virus production machine, releases more viruses into the body, and you start getting ill, and you're starting to fight off that. If you prepare your body, the aim of a vaccine is to prepare your body for that. And so when it comes in, then your body can realise, oh, hang on, this is just a COVID thing that I've seen before, and can fight it off really easily without having to work out what it is first and then kind of assess how to destroy it and then destroy it. And so one of the things that this vaccine does, especially the mRNA vaccine, which is this kind of new technology vaccine that people kind of worried about because we've never had it before, but actually there's nothing to worry about because we're not injecting anything dangerous into you. Just a bit of mRNA, and that's the really safe bit of it. So the mRNA is just a bit of RNA that's going to tell your cell to make a protein. And the bit of the protein, the bit of the cell that does that is the ribosomes. And so if we look back up here, we've got ribosomes in our animal cells, these tiny little flecks of things. They're going to make these proteins. Oh, there we go. They're going to make these proteins, and then your body's going to recognize these proteins and be like, hang on, these aren't proteins anything to do with us. We can destroy them. But the thing is, these proteins are similar to a COVID protein, and so your body can destroy these proteins really easily. But then when the actual virus comes along, it recognizes those proteins and thinks, hang on, I've dealt with this before. I've smashed these proteins up. I know how to deal with them, and so then it can destroy the whole virus and mean that actually infection and the dangerousness of having something like COVID-19 is really lessened because your body just like, oh great, I know how to deal with this, done, out of the way, what's my next task? And so the mRNA is really important to be able to get that protein in a safe way to make your body make it, and then you can just deal with it and it works. So it's not trying to take over your body. It's not something that's going to last a long time. This mRNA, your body makes its own mRNA from DNA in order to make a protein. So we talked about the protein hemoglobin. Actually, in order to make hemoglobin, we make some mRNA, and then we make some hemoglobin from that mRNA. And so we're just using some different mRNA to be able to do that. Obviously, if this mRNA stuck around for ages, then it would make the protein again and again and again. So actually, mRNA by nature breaks down quite easily, and it breaks down easily, whereas DNA, that is the blueprint of this, doesn't break down very easily at all. So it's pretty safe to inject it into your body, and hopefully it will give you protection, and that's a great thing. So bring on the vaccine, that's what I say. Right, let's look at another couple of specialised cells. So we've done a red blood cell, let's talk about a nerve cell. So I've mentioned nerve cells before in my videos, so do have a look and check them out if you want to. There's plenty there. The nerve cell has this kind of end to it, and then it has a long body, and then it has a few more things at each end as well. And so this kind of star-shaped thing on the left-hand side, this is where the nucleus is. This one does have a nucleus, and this is the cell body. And so that's where most of the cell processes are. That's where we've got our cell membrane, we've got cytoplasm, that sort of thing. The really long structure is an axon. And then at each end, we've got these things that are dendrites. And a dendrite is just something where it can make a connection with a different cell. And so because it's so long, and because it's got these dendrites, it's able to make connections with different cells and be able to pass on these different signals from one cell to another, from one nerve cell to another. The other thing that's really important is in its cell body it has lots of mitochondria, and they are able to help it generate the signal. So in order to make a signal, it needs to send lots of proteins going and make sure that they're turning things like ions in and out, in and out, in and out. And so in order to get them to go in and out, we have lots of mitochondria that do all of that energy-ness to be able to sort it out. Fantastic. Let's talk about a couple of plant specialised cells, and then we'll do differentiation, microscopy, and then we'll aim to end about 45 minutes, maybe more, like an hour. So next thing is a plant cell. Plants are green. Let's go for a green pen, and we're going to talk about root hair cells. Now it's good to differentiate and say that these are plant cells, because sometimes we think of root hair cells. Oh, hang on, I've got hair, and then the roots of my hair. So maybe these are cells at the roots of my hair. But instead, it's the other way around. Plants have roots, and on their roots they have hairs. And so these are the cells that are on the roots of a plant. And so they look a little bit like this. Remember, a plant cell, we've got a square structure, but this time they have something poking out of it as well. And so they'll have a large vacuole that kind of mimics that shape as well. They'll have their nucleus, they'll have a cell wall that will go all the way around. And their purpose is all about moving water and mineral ions into the cell. And so they've got some features that are going to help them do that. So they've got this large surface area because they've got this hair on them. So a large surface area is going to mean that lots and lots of water can move in. They also have mitochondria in them. That's going to give them the energy to move in, and so the energy can be able to pump in things that won't just move in naturally. So for example, mineral ions, so potassium in the soil, and there's lots of potassium already in the plant and not much in soil, and so it needs to pump it in through active transport. Simon's added that it keeps it turgid, the perfect state. Yeah, so when we talked about our plant cell, we talked about how it's got this vacuole in the middle, and that's going to fill up, and then the cell membrane is held in place by the cell wall. So the cell wall means the cell won't burst. So it can be turgid and basically strong, so it can hold the plant upright. It's important to note, animal cells can't be turgid because they don't have cell walls. So if our cells, if you put a red blood cell in pure water, it would end up just bursting because it hasn't got a cell wall to hold it in place. Now, let's look at a different one. Let's look at a xylem cell. Again, another little simple plant cell. This one is one that's going to transport water up the plant. And actually, it's a cell that's died, unfortunately, but it's got these rings of lignin in the middle of it. So this cell, what happens is it grows and then it dies. And once it's grown in this very long, straight shape, it then dies, and this lignin in the middle forms and holds it in this open shape. It dies so that water can just carry on moving up in a continuous column. It's not going to, it's not going to be interrupted or anything like that. It can go all the way from the roots all the way up to the leaves without stopping. And so the xylem cells, they've died on purpose, and then this lignin holds them in place. Now, what is interesting is that you might have heard, it might be something like your granny maybe once told you that if you count the rings on the inside of a tree trunk once it's been chopped down, then it will tell you exactly how old the tree is. And you kind of think, oh, isn't that a nice fact? It's probably not even true. It is true, and let me explain why. So inside a tree trunk, you might chop it down. Every layer you see there was a layer that for a season, just near the edge of the bark, was a xylem cell. And as it died at the end of the season, that xylem cell collapsed because the leaves have fallen down. It doesn't need to pump water up if there's not any leaves in there. So the xylem cell collapses, and a new xylem cell then grows beyond that one. And that first one that has just died gets pushed in to the centre of the tree. So every season it goes through, every time that the leaves fall off, which happens to be once a year, then it forms a ring. And so it is actually true. And if you count the rings from the centre to the outside, that is how many years the tree has been alive, which is pretty crazy, but also a really cool way of finding out about trees. So next time you see a tree stump or something like that, see if you can have a count. I think it's a bit crazy that something that was alive for that long is now a log. Cool. Let's carry on a little bit and talk about cell differentiation. So I'm going to say that is cell specialisation tick. We've talked about red blood cells, nerve cells, and two types of plant cells as well. And so if we go to cell differentiation, let's do orange. then that is something different again. So cell differentiation is all about how cells turn from a stem cell into something different. So they're going to change from a stem cell into something better. Now let's explain what a stem cell is. A stem cell is a cell that can become any type of cell. And that might be any type of cell, literally, it could be anything at all, or it could be any type of cell within a limit of different types of cells that are on offer. And so you get stem cells in an embryo, in an unborn baby, where they can literally turn into any type of cell. You take a cell from a baby and it can just turn into a liver cell, it can turn into a brain cell, it can turn into a foot skin cell, or a muscle cell. You can get other stem cells that don't quite work in that same way, that actually work in adults, and they can differentiate into a few different things. So you can get a blood stem cell in the bone marrow that can turn into lots of different types of blood cells. So you can turn into a red blood cell, a white blood cell, or a different type of white blood cell. But a stem cell basically can turn from something that's quite broad into something very specialised. So they turn from stem cells to specialised cells. And that's really useful in your body, because it means that your body can end up turning some parts of the body that it needs into something else. In plants, they can do this throughout their whole lives. So you might have seen someone planting like a willow arch, and it's kind of got these willow trees arching over each other like that. This isn't going to make sense if you just look at the PDF later and you haven't watched the video. But what they've done there is they've just got some trees, and the willow branches, they can chop them off, sit them in the ground, and this willow bit that was part of a branch can suddenly differentiate, become different, and decide, actually, I need to be a root now. I'm surrounded in soil, there's lots of water here, I need to get it inside. And so it starts to grow root cells, and it starts to put down roots, and it actually turns into a new willow tree from just those plants that have been chopped off and put in different parts of the ground. Now, obviously, I can't chop off my finger, put it in some water, and hope it will turn into a new human being. That's not how reproduction works. But there are parts of your body that can just grow back. So your skin cells, you might have probably got a graze or a cut or something like that. And so that sort of thing, it's just basically some skin cells grow, they divide, they can fill in that gap, and they can heal over that thing. Goodbye, Simon. Thank you for watching. And hello to everyone else who's just joined. I see the viewer count has gone up, so it's great to have you with us. Now, finally, I think that's enough on cell differentiation. There's plenty more to say, but I want to make sure we don't run on too long. So let's lastly look at some microscopy. So microscopy is all about seeing small things. What colour should go? Let's get purple. Seeing small things. And so light microscopy has been around for hundreds of years. Basically, as soon as we managed to start making lenses and glass properly, then we were able to make microscopes and to be able to really narrow down and look at small things. And actually, as technology has improved, we've got light microscopes, but we've also got electron microscopes. So we can look at things through looking at a beam of light that goes straight into our eyes that we can see down a microscope, something you've probably used in a classroom at some stage in your life. And then we have electron microscopes, probably something that you haven't seen in your life and probably haven't looked at. If you have, then amazing, that's really cool. And they are fascinating pieces of equipment, but they are very expensive and difficult to use. So let's talk a little bit about how they work and the advantages and disadvantages. So we'll have two columns. You know, this is the kind of thing that I'd say draw a table neatly with a ruler in a class, but you know what? We're on YouTube, and so we can write without rulers. So we're going to have a light microscope, electron microscope, advantages, disadvantages. So advantages of a light microscope is that they're really easy to use and they're really cheap. So actually, you know, you can have a load of good quality ones in your classroom and just everyone can see something at once. You can see something quickly as well, and you can see things that are smaller than the human eye would normally be able to see. But the negatives is that the resolution is limited and the magnification is limited as well. And so you can see things, but as you go in too much, if you zoom in too much, so if you magnify an image too much, it starts to become a little bit blurry. It almost, you'd feel like it would be pixelated in this modern era, but it's not pixelated. It's just that actually it's something, you're looking at something smaller than the wavelength of light. And if it's smaller than the wavelength of light, a light microscope can't see it. And so that's where we end up being limited, and why we can't see things like, so if we think about a cell again, I can probably see a nucleus, I can't see mitochondria with a light microscope, because it's just, the mitochondria are just too small. Whereas with an electron microscope, easily see a mitochondria if I want to have set it all up. So electron microscopes, the disadvantage of those is that they're really expensive, and preparation of a slide takes ages. So if I wanted to, say a long time, maybe sounds a little bit more formal, doesn't it? If I want to prepare a slide for a light microscope, then I can do something like get scraping from the inside of my cheek, I can peel a little bit of an onion, I can put it on a slide, put a little bit of stain on it, maybe some iodine, some methyl blue, and that basically means that the colours stand out a little bit more. The stain might bind to parts of the cell that we want to see, so it might basically hang on to the nucleus, cell wall, cell membrane, so I can see those bits a bit more clearly. And then I look down the microscope and it's there, and I can do that in 10 minutes. You can fit it into a half-hour lesson basically. But with an electron microscope, this is something that takes a lot more time. So you've got to prepare something. It often needs to be frozen. It can take weeks to prepare a slide, and then you put it under your microscope and you can't even see it. And that's the crazy thing, is that obviously we can't see electrons. If we could, then that would be a completely different way of life. But you can't see the electrons that you're firing at this thing, and so you have an electron-sensitive thing that will be able to detect the electrons that bounce off in different directions, and then you can finally put together this picture of a subject. Now, someone's just asked again if the live stream will be archived. It's okay, I'm going to leave it available so everyone can see, so you can catch up from the beginning, and I'm also going to make the PDF available too of these notes, so you can have fun trying to decipher my handwriting afterwards. So don't worry, it'll all be there, so you can watch back. But thank you for your question. Now, there must be some disadvantages of the electron microscope. There must be some advantages of the electron microscope, sorry. We know about the disadvantages. And the advantages is that we can magnify at much greater magnifications than the light microscope, and we can also do a much greater resolution. We can see tiny little things. Now, there are actually two types of electron microscope, two broad types, that is. So we've got transmission and scanning. So transmission electron microscopes, they send electrons through things, and so you end up with a 2D image, and that's really useful for seeing inside cells. We can fire electrons through a cell, and we can see all those different parts. And then the other type is scanning. And so if you Google scanning electron microscope images, or SEM images, then you see some amazing things. These kind of 3D structures, because what they've done is they've sent us scanning electrons over the surface of something. So rather than try and fire them through a very, very thin sample, maybe a thicker sample, and you can see the whole structure. So you can see things like a flea or a louse and all the individual details and hairs of them. Sometimes a bit gross, but pretty cool. So that's a scanning electron microscope, and you get 3D images with them, and they see the outside of things. And that's a 3D image. Great. Now, the last thing that someone just asked if it was like an MRI scan. So MRI scan is slightly different. So they pick up images in a similar way through X-rays, but the way that MRI works is that there's magnetic resonance imaging. So every molecule in your body, basically, every water molecule in your body has a slight polarity. So there's a slightly positive side and a slightly negative side. And so what you can do is you can use some powerful magnets to turn all the molecules so they're all facing one way, with the negative end one way and the positive end the other way. and then as you move and as your brain uses stuff, then those molecules start turning around, and so you can pick up on those little turns and those little detections. So that's really, really interesting, basically. So that's how MRIs work, but it's different from microscopy. Now, the last thing I thought we'd do is we'd do a couple of calculations of things like magnification. So if we are told the magnification of something, then we can work out the size of the image we had beforehand, or the size of the actual real-life thing. So the magnification is equal to the image divided by the actual size. So, for example, if I gave you an image and said, "This is an image of a cell," there you go, but very basic cell, and I say this cell, I've measured it, after I've magnified it, so I've magnified it, let's say 50 times, and this cell is now 3 centimeters long. Then I can work out exactly how big the cell was the actual cell was. After it's magnified, it's 3 centimeters, great, but how big was it beforehand? And so I then put it into my equation here. So you might have used a triangle like this to do an equation. I'm not sure whether you've done that before. I find it really helpful because we can end up doing three different equations in one. So we cover over the one that we want to find out. So we know what the magnification is. We've said that's 50 times. And we know what the size of the image is after it's been magnified. We've said that's 3 centimeters. And so then we can work out actual size over here by covering this one up. And then if it's like that, then we divide it down, or we think of it I over M, and that works as well. And the way I kind of remember this is sometimes that you can think of someone called Mia who likes microscopes, and there you go, that's Mia, and that's a little magnification triangle. But anyway, let's go back to this calculation. Whoops. So we've got there 3 centimeters, and we're going to divide it by 50. 3 divided by 50 is what? Let me get my calculator up, because you always need a calculator in science, and mental maths will get you somewhere, but not everywhere. And it is 0.06, and that means that this cell in real life is 0.06 centimeters. And you might want to change it into millimeters and say that it's 6.6 of a millimeter. So it's pretty small. But that is just one way that we can use this equation. Now, if we go back and we say MIA again, and this time we are given the magnification size. We are saying that something is magnified 200 times, and its starting image was maybe 1 micrometer. We can then work out what the image is, so we cover it over and see what we've got left, MA at the bottom, so we times them across, and so we say actually the image will end up being 200 micrometers large. And so yeah, Jasper's absolutely right, you can make this kind of pyramid with lots of different scientific equations, only ones that are with 3 things on, but it's pretty cool. A couple of other questions. Ruben's asked, can we see things as small as electrons? Not quite. We can see things as small as the wavelength of electrons. In the same way that we can't see photons, like the light particles, with a light microscope, but we can see things that are as small as the wavelength of light. So we can actually, with some types of electron microscopes, like a tunneling electron microscope, you can see things that are like actual atoms. So we can see atoms and their arrangement, which is really cool. So that is worth it. Someone says, I think that's right. Excellent. I'm glad people agree. People are getting mixed up with equations like this one. So like speed, distance, time. It says time as well. Yep, so if we've got speed, distance, time, then we have speed, distance, and time. And we just need to cover over, we can fill in what the symbols would be. And we always divide it down, and then we have times there. And all we do is we just cover over the one that we don't need. So if we cover over time, then it's going to be distance divided by speed. If we cover over speed, then it's going to be distance divided by time. And if we cover over distance, then we do speed times time. And it's just a way of working out all those different things. If we're giving 2 parts of information, we can always find out the other one quite easily. I'll tell you that, that's physics, you don't see physics. We can live without physics, even though some people have to learn it and I have to teach it sometimes. Fantastic. Let's go back to the top and just tick off and say we've done microscopy as well. Fantastic. Thank you so much, guys, for sticking with me. That is 50 minutes of learning there. Really appreciate the comments and appreciate you guys watching. As I said before, I'll leave the video up, and so you can always watch back and pause it and things like that and check your knowledge. Do carry on putting questions and things in the comments. Do subscribe, and then you'll be able to see my next live lesson. We'll try and do it at the same time next week. It's always good to have a bit of consistency. That's why teachers love a timetable. But it would be great to see you there as well. We'll carry on going through the course. I'll let you know what topic that is. I need to look at the specification and work out what that is. I'll put a link to the PDF in the description in a moment. I just need to process it, and I might add a few more details of things that I've said but not written down as well. But I'll make sure I put that up there. But as I said, thanks so much for watching, and yeah, I look forward to seeing you next time. Thanks so much. Have a good evening.